All right, let's get right into it. Today, we're going to break down some really fascinating new research that's introducing a whole new way to think about fighting cancer. Forget the old sledgehammer approach. This is more like a microscopic smart bomb designed for one and only one mission.
This is the big question, right? It's been the central challenge in cancer therapy for decades. How do you possibly wipe out the bad guys without causing a ton of collateral damage to all the healthy cells around them? It's the problem that has really defined the limits of our best treatments.
When you're talking about blood cancers, things like lymphoma and leukemia, the standard approach has often been that sledgehammer we mentioned. Chemo and other drugs, they basically attack any cell that's dividing quickly. And yeah, that hits the cancer, but it also hits a lot of your healthy tissue. That's where all the awful side effects come from. And even worse, sometimes the cancer cells just learn. They become resistant and the treatment stops working. We've got to find a better way.
So if you're going to build a smart bomb, what's the very first thing you need? You need a target, something totally unique to the enemy that your bomb can lock onto. And that's exactly what these researchers set out to find. A special kind of flag that only the cancer cells are waving.
It turns out that flag is a protein called LFA1. Now, pretty much all of our immune cells have this protein, but here's the absolute key. In these specific blood cancer cells, that LFA1 protein gets jammed. It gets stuck in a permanently on position. So it's basically like a glowing beacon that's just screaming, "Hey, I'm a cancer cell over here." And here is the visual proof. It's so clear. If you look at that chart on the left, you see that huge peak? That is showing the high level of that active, glowing LFA1 flag on the malignant blood cells. Now, just glance over to the chart on the right. That's the healthy cells. The peak is practically gone. The flag is down. This is our bullseye. It's a clear, reliable difference between the bad cells and the good ones.
Okay, target acquired. Now for the fun part. We have to actually build the weapon. And this isn't some drug that just floods your entire system. No, this is a precision delivery system engineered from the ground up to seek and destroy. The best way to think about it is like we're building a microscopic drone. So step one, you build the shell. It's a tiny little sphere called a nanoliposome, and this is what's going to hold our payload. Step two, you give it a homing device. You coat that shell with a special antibody that acts like a heat-seeking missile, and it only looks for that active LFA1 flag. And finally, step three, you load the drone with its cargo, a very special set of instructions designed to shut the cancer cell down from the inside out.
And here it is. This is what the whole thing looks like when it's put together. You can see all the parts we just talked about, the outer shell, those little homing antibodies studded all over the surface, and tucked inside that precious cargo ready to be delivered. It's just a really amazing piece of nano-engineering.
So what is this drone actually delivering? It's not a poison or a chemo drug. It's something way more sophisticated. It's a tiny piece of genetic code that's designed to disarm the cancer cell's survival machinery.
The cargo is a molecule called siRNA. Think of it as short interfering RNA. And its mission is incredibly specific. Get inside the cancer cell, find one particular gene, and just turn it off. The name of that target gene is WASP.
So why WASP? Why go after that one? Well, because this protein is basically a master accomplice for cancer. It helps the cancer cells survive when they shouldn't. It gives them the ability to move around and migrate all over the body, and it lets them invade healthy tissue. In fact, studies show that the more WASP a patient has, the worse their prognosis is. So taking it out of the equation is a really critical blow.
The whole theory is just beautiful, right? You've got a unique flag, a smart bomb, and a crippling cargo. But does any of it actually work? Let's get to the evidence and see what happened in the lab.
This slide just demonstrates that incredible precision. It's amazing. The researchers mixed cancer cells and healthy cells together in a dish. Then they added their nanodrones, which they tagged to glow bright red. And look what happens. The red drones are just gobbled up by the larger cancer cells, while the smaller, healthy cells are left completely alone. It hits the target and nothing else. Perfect.
And what happens after that cargo is delivered? Well, this chart tells the story. One of the deadliest things cancer cells do is move around. So before the treatment, you can see these cells were migrating all over the place. But after the treatment, with the WASP gene silenced, their movement was almost completely shut down. They were essentially paralyzed, stopped in their track.
Okay, all of this is incredibly promising in a lab dish. But let's be real. The ultimate test, the question that truly matters, is what happens in a living organism. Could this system actually find and shrink tumors in a living mouse model? And the answer is a huge, resounding yes. Just take a look at this graph. The black line, that's the untreated mice, shows tumors just growing completely out of control. But the blue line, that's the mice treated with our smart bomb. It's nearly flat. Tumor growth was dramatically, significantly suppressed. The treatment was absolutely working.
Let's put a hard number on that success. By the end of the experiment, the tumors in the mice that got the treatment were 4 times smaller than the ones in the untreated group. A 4-fold reduction. That's a massive, massive effect.
And this is where we come full circle. Remember that sledgehammer problem from the beginning? Well, the researchers watched these mice very closely and found no signs of systemic toxicity. No weight loss, no damage to healthy organs, nothing. The smart bomb hit its target without causing all that collateral damage. This is the precision we've all been waiting for.
So what does all this really mean for the future? This breakthrough, it's actually much bigger than just one new potential drug for blood cancer.
You see, the most important takeaway here is that the researchers didn't just create a single drug, they created a platform. It's a modular system, like building blocks. You can change the homing device on the outside to seek out a totally different cancer flag, or you could swap out the cargo on the inside to deliver a completely different set of instructions. It's a true template for precision medicine.
And that leaves us with this final, really powerful question. This technology was built to target one specific flag on one type of blood cancer. But what happens when we start identifying other unique flags on other types of cancer? Or maybe even on cells that cause autoimmune diseases? The potential to adapt this smart bomb platform for all sorts of other missions, well, it's truly immense.